So, a couple of things to give you a heads up of things that are coming up in the next couple of weeks. For lab, we're transitioning into those independent projects. So tomorrow is going to be a day where I want you to take a deeper dive into the topic that you're researching, begin to find some articles if you haven't yet, and also to begin to come up with focus in on a question, a very specific question. What is your hypothesis? What is your prediction? And we'll start to walk through that together tomorrow. So you'll have a couple of assignments coming up, and I need to double check the due dates. One of them will be laying out for me what's the question, what's the hypothesis, what's your prediction. And then the second one is an annotated bibliography of articles that will help you with the research that you're doing. So that's one thing that's coming up. The other thing, sorry, this is not comfortable. I will have the exams to you tomorrow. I forgot them in my office, but I do have for you the sheets from your videos. So I'll give those back to you guys today as well, okay? Overall, I was really happy with them. I thought there was a lot of good work done. So you guys should be pleased with what you did. I thought they were really good. And then last thing, because I feel like I'm going to forget this if I don't mention it while it's in my head. So next week, Tuesday, 6 o'clock, I think, in Washburn, I'm giving a 2-hour talk. It's not just me talking 2 hours. But it's me talking about, as some of you know, my son Jake is trans male, so I'm going to be talking about him for a while. This actually came out of a conversation that happened last semester. A couple of people, including Vincent Martinez, asked if I would be willing to talk about our family, because, especially for folks who are going into the medical profession, Madison is kind of a hotbed for transgender health. So I'm going to be talking about that for a couple of hours. So if you want to come, no, I've heard there's been questions about if there's extra credit attached to this. No, not to this one. Because I don't want to obligate anybody to have to come and listen to me for 2 hours. But just wanted to let you guys know of that ahead of time. All right. I figured we'd start with multiple choice questions, since this is something that some of you have suggested we should try to do some more of. So at least for today and Wednesday, I've got a couple for us, one to begin with and then one to follow up with a little bit later. You don't have to log on to Socrative to do this. We can just do it here without you having to report any answers. But for this one, the question reads, which of the following descriptions best describes the central dogma? And we're going to be spending the next 4 days talking about the connection of molecular biology with genetics. And we will talk a little bit later about how does this connect to themes like antibiotic resistance. So which of these best describes the central dogma? Is it A, DNA is translated into RNA and RNA is transcribed into protein? B, RNA is transcribed into DNA and DNA is translated into protein? C, RNA is translated into DNA and DNA is transcribed into protein? Or D, DNA is transcribed into RNA and RNA is translated into protein. So think for a minute. Check with the partner. I'm a little disappointed there's not.
I know. We said it before we got here.
It's just really disappointing. All right, which one is the correct answer?
D.
D. All right. So a couple of key things to keep in mind here is just what is the connection between DNA and RNA and protein with respect to the central dogma? And really what we're talking about is flow of genetic information. Okay? So the process of DNA making more DNA is known as what? It's not one of the terms up here, but DNA just making more DNA is what? That's our replication. DNA to RNA is translation. Where do these events occur in a eukaryotic cell? So where does replication occur in a eukaryotic cell? Nucleus. How about transcription? Nucleus still. Translation? That's in the cytoplasm. Where do these occur in a prokaryotic cell? Everything's in the cytoplasm, since there is no nucleus. And that creates a couple of unique features. It also creates a couple of differences between prokaryotes and eukaryotes. So today's main focus isn't so much on these three processes. I think in part this is a bit of a review for some of you. It's more what's unique about these processes in prokaryotes versus eukaryotes. So the first one is location. Because prokaryotes have no nucleus, all of these events are taking place in the cytoplasm. One of the unique things that creates then is that transcription and translation can actually occur at the same exact time on an mRNA. We'll see a picture of this a little bit later. All right. To give you a heads up of where we are going in the next few days with this. Today, just the basics. We'll talk about the process of the central dogma. So the replication, transcription, and translation. I want to make sure that we have a bit of time to review the homework and to kind of group terminology together. We'll talk about how prokaryotes are unique and some of the structural differences between prokaryotes and eukaryotes in the central dogma. And then we just want to spend a little bit of time talking about how is this genetic information organized in bacteria. Wednesday, we'll talk about transcribing DNA into RNA and then RNA into amino acids, the translation process. We'll also talk about mutations. And then Friday, we discuss gene expression, and the following Monday, we talk about horizontal gene transfer, which is a process by which organisms can exchange genetic information between them. What we're talking about here, one cell replicating its DNA to produce two daughter cells, that's what we call vertical gene transfer. So it's from mother to daughter cells. And that is different between to horizontal, which is between two cells that don't actually have to undergo division to exchange genetic material. All right, let's for 5 or 10 minutes, roughly, what I want you to do is take some time with your concept maps if you have them, or try to memorize what you can of it, or if you have it on your computer or something. And with a partner or a couple of individuals, what I want you to do is work on helping to solidify some of those connections between different terms. For example, there were a couple of times where I saw sigma factor associated with translation. I don't think that's the best place for it. So I may have put a couple of comments on the homework assignment, but what I want you to do is check it over with somebody else. And if you have questions, let's make sure to address those in a large group once we report out. So go ahead, take about 5-10 minutes. I'll be walking around to help out as well. Anybody have any questions about any of the terminology? Annie?
The sigma factor, is it on the RNA polymerase or the ribosome?
Yeah, it's on the RNA polymerase. So if we look at, so Annie's asking about what's the sigma factor, and I liken the sigma factor kind of to like it looks like a kidney bean in some cases. So the sigma factor is part of what we call the holoenzyme. And this is more specific than what you need to know, but the holoenzyme refers to the actual functional unit of the RNA polymerase and the sigma factor. The functional unit, this larger piece here, represents what we call the core polymerase. So when we talk about RNA polymerase actually transcribing, it's that core segment that performs it. What is the function of the sigma factor, though? What's that?
To initiate transcription.
It does initiate, and how does it initiate?
Binding to the promoter.
Good, binding to the promoter. If we wanted to be even more specific than that, what we would say is it binds to a very specific sequence in the promoter. So there are, in E. coli, 6 or 7 different sigma factors. And each one of them binds to different regions that are about 35 nucleotides before the transcriptional start site, and about 10 nucleotides before that transcriptional start site. I refer to these just as the -35 and -10. Sometimes that -10 is referred to as the Pribnow box. But the role of the sigma factor is just to bring the RNA polymerase to that site where transcription is going to be started. Okay. For each transcriptional event of mRNA in prokaryotes, the same exact RNA polymerase is used. But it's those different sigma factors that are used to direct that core polymerase to different sets of genes that are going to be turned on or turned off during periods of stress, or, for example, high heat versus normal temperature, for example. Does that answer the question, Annie? Okay. Other questions? Okay. What's the function of gyrase? It is a target for antibiotics. That's not necessarily its function, though, right? We will come back to it as a target for antibiotics, and I think, Emma, you pointed out quinolones are, right? And that's something that I brought up. We will come back to that when we talk about antibiotics. But that's not its main function. What's the main function of gyrase?
Doesn't it separate the DNA?
It does help to separate, but it's not the same function as helicase. So helicase is the one that does what? Unzips, yeah, unzips or separates. If we were to look at if we were to look at helicase, so helicase is the one that is actually moving from that origin of replication, where replication begins, and is moving down the double strand, and it's just breaking the hydrogen bonds that exist between AT base pairs and GC base pairs. Gyrase's function is a little bit different. So imagine if, I always forget to bring a model for this one, but imagine if you had a rope, a typical braided rope that is kind of twisted on itself. And if you were to take those braids and start to separate them from the middle, what happens on the ends? It gets so coiled, and that's exactly what happens to DNA. Is the number of coils remains the same, but the area that they take up is much smaller, and that produces what we call supercoiling. So the role of gyrase is to minimize that supercoiling that gets formed. And the way that gyrase does that, in simple terms, is, I'll draw this out if I can. I'm going to draw my 2 strands of DNA different colors here to make life easier. What gyrase will do is it will cut one of those 2 strands and actually allow for it to loosen tension. It allows for it to uncoil, and then after that uncoiling has occurred, the gyrase will paste those that backbone back together. And when we say that it cuts it, what we're really meaning is that it cuts between a sugar and a phosphate that make up the backbone of DNA. Okay? So that's one function. There's another function that is more specific to prokaryotes than it is to eukaryotes. What's the nature of the E. coli or bacterial genome? How is it structured? It's circular. And so one of the key issues that happens is that those circular genomes, they become linked. And to separate those obviously, if they're linked, they cannot move to opposite ends of the cell so that a cell can replicate and produce two daughter cells. That's where gyrase comes in again. It'll actually cut one of these two, both sides of the backbone in this case, allow for those two strands to separate from each other and then paste it back together. Emma.
That's in eukaryotic?
This is prokaryotic. Actually, it would be the case whenever you have a circular genome like this. It's just more common in prokaryotes to have a circular genome than it is in eukaryotes. So this would happen with plasmids as well, not only with the genome, but also with plasmids. Now to Emma's point, she brought up that gyrase is a target of antibiotics. And one of the antibiotics is called nalidixic acid. I'm just going to draw a quick picture to show you what happens. Oops. Okay. So what we find is that typically in a bacterial cell, we can actually stain cells to identify the membrane and then identify the nucleus. We can use dyes that stain the nucleus a nice blue color, so we can, or sorry, not nucleus, wrong term, genome, right? It will stain the chromosome, and that's where we identify where it's at. When these cells divide, normally what we find is that the cell will elongate, and over time, these chromosomes end up moving to opposite ends of the cell to produce two new daughter cells, each with an identical copy of the genome, theoretically. Now, if we apply nalidixic acid or one of these gyrase inhibitors, what we find is the following. Instead of getting the separation of the DNA, as we see up here in the right-hand corner, what we find is these cells elongate, but we still get one region where the DNA is located. It's because they're not actually physically separating from each other. And these cells will continue to grow in length, and yet their replication or ability to separate those genomes is halted because, one, the ability to resolve supercoils has been inhibited, and then two, the ability to separate those chromosomes has been inhibited as well. And so for drugs like nalidixic acid, their mechanism to blocking cellular growth is just block the replication process. These cells are still viable, they're still alive, they just no longer can grow. So we would call a drug like this a bacteriostatic drug, or one that inhibits the growth of the microorganism. What does a eukaryotic mRNA look like? So I'm sure at some point all of you have been taught what basic characteristics of an mRNA are. What are the basic characteristics of an mRNA? Let's start with this. What bases do they have? They do have codons. Let's go back even further, though. They have AGs and what else? Cs. What's the last one they have? Do they have Ts? RNAs don't. They have Us, right? AG, Cs, and Us. That's the first distinguisher. The other things that we find with eukaryotic mRNAs is that they've got, well, we kind of draw them like this. What's this feature here, and what's this feature here? And this is the 5' end, this is the 3' end. So what's at the 5' end? Usually there's some sort of cap. All right, so usually it's a methylated guanine. That's not important. All you need to know is there's a cap there. And that cap is to protect that RNA from degradation. What's on the 3' end? Apparently we found where we need to spend some time. Poly-A tail, or polyadenylation tail. Okay. Basic structure of a eukaryotic mRNA, like this one here, is it's got a 5' cap and it has a polyadenylated tail. These are features that are unique to eukaryotic mRNAs. We do not find these in prokaryotic mRNAs. In prokaryotes, oftentimes we find no cap whatsoever. And instead of a poly-A tail, we find sometimes what's known as a hairpin loop structure, or what's known as a termination factor of transcription. Basically, what happens is during the transcriptional process, the ribosome, the RNA polymerase will transcribe this region. This hairpin loop forms, and this is basically base pairing between the RNA itself. And when it does that, it basically destabilizes the RNA polymerase and knocks it off from transcribing any further. Okay. So these are some basic characteristics. There's another characteristic that distinguishes eukaryotic and prokaryotic mRNAs from each other. Again, eukaryotic mRNAs, like human RNAs, are made in the nucleus. Before they can be exported from the nucleus, sometimes there are regions of the RNA that have to be cut out and removed to form the mature mRNA. What are those regions that are cut out called? Sometimes we draw them like this. So this would be the region that's cut out. Shall we play hangman? Two words. One for this structure and one for this structure. That was a horrible start. Man, this is the longest we've played yet this semester. Oh, thank God. Okay, somebody read this one. Intron, okay? What's this one then? Exon, okay? So there are introns and there are exons, all right? Exons are, so the introns are the regions that get removed. So before a eukaryotic mRNA makes its way out of the nucleus, basically it goes through a modification process. The introns get cut out, the exons get pasted together, and so all that's leaving the nucleus are the exons that actually contain the region of the mRNA that encodes for a protein. Now, prokaryotes, do they have these? Why not? I suppose why not's not the right question, but they don't have them. And in part, you could argue that there's no evolutionary selection for them. But again, transcription and translation can occur at the same time. And so that means that the mRNA, as it's being transcribed by RNA polymerase, there's not going to be any sequence removed, and all that's going to happen is that mRNA is going to feed right into a ribosome. What we find is that let's see if I can find this real quick. In prokaryotes, transcription and translation can occur at the same exact time. So that as transcription is occurring, within a few seconds after it's starting, an mRNA can be read by a ribosome and be translated as well. This is unique to prokaryotes. And again, the lack of a nucleus allows for these two things to happen at the same time. Also, the lack of introns allows for these two things to happen at the same time. One of the reasons why I bring this up is not only to contrast the two to each other, but to show that this actually has a lot of power. In prokaryotes, in a eukaryote, oftentimes we have to have transcription to occur, the mRNA is modified, the mRNA exits to from the nucleus into the cytoplasm, and then it's translated. Would this process be quicker or slower that we find in prokaryotes? So to go from the beginning of transcription to protein being made, is this faster or slower? Yeah, it's much faster. We can find that from the point of transcription being initiated, we can detect a protein from an mRNA within 2 minutes in prokaryotic cells. In eukaryotes, we'd be talking about 10, 15, 20 minutes for these events to take place. And so regulation of genes in prokaryotes and eukaryotes is slightly different from one another. One of the reasons for that is since prokaryotes lack a nucleus, they can turn on and turn off genes incredibly fast. And that's quite different from what we find in eukaryotes. These structures are sometimes referred to as Christmas trees, because you've got, this would be like the trunk of the tree, and then each of these represents a branch. So you've got your RNA polymerase, your mRNA is the branch, and then your ribosomes are like the ornaments on the Christmas tree. At least that's how it's been described in the past. And then multiple ribosomes like this is sometimes referred to as a polysome, though that's not a term I'm requiring you to know. Okay. How does translation occur? What's required for translation? So I'm going to give you guys 2 minutes. What I want you to do is just write down a list. Everything that's involved in translation. Not what they do, just what are things that are required for translation. Go. Translation. All right, what's required?
mRNA.
That's a good start. mRNA. What else? Ribosomal subunits. Can I just put ribosomes for now? Okay. What's the ribosome made of? What's that? No, what was that? That is true. Ribosomal RNA and protein. So this is not just a protein structure. There's some RNA in there that's involved in the process as well. What else? All forms of RNA are involved in this process. Absolutely. Other things that are needed. We need amino acids. Those obviously are going to be involved in making our polypeptide or protein. Those are the major things. Of course, we require some form of energy, right? That's going to come in the form, typically with GTP here. Which of these contains a codon? mRNA contains a codon. What is a codon? 3 nucleotides that encode for an amino acid, right? So if we have, for example, the start codon, AUG, not only does it start the process of translation, but this also encodes for an amino acid. AUG is in the 5' to 3' direction. What has the anticodon sequence? Our tRNA. Beautiful. Alexi. Hey, how are you doing?
I'm good, how are you?
Good. How was break?
Feels fine.
Good. What's the anticodon to AUG?
Would be U. A, C.
Okay. Which end is over here? 5' or 3'?
3.
Very good. All right. So our tRNA would have this as the anticodon, and then it's got a 3-dimensional structure. Something like this, often described as a clover leaf. And let's see, 3, 5. Right here would be our amino acid methionine, or MET, sometimes abbreviated. Where does this base pairing, where on a ribosome, in which of the 3 sites of a ribosome does this base pairing actually occur? So there are 3 sites in a ribosome, the E, the A, the P, and the A site. What do those different terms mean? Or what happens at each of those 3 sites, maybe? It's a different way to ask this question. So what's happening here at the A site? We've got a codon GAU. What's happening here? What's that?
Release doesn't matter. Or is it binding there?
Not the amino acid, but the tRNA is binding there at this site. A I usually think of as like acceptor site. I don't know if that's exactly what it stands for, but that's the first thing that comes to my head. So this is where tRNA is coming and binding to the mRNA. If it doesn't match up, the tRNA exits. If it matches, we have a stable interaction. Ribosome then will, at the P site, the P stands for peptide. What happens is we get a bond between the amino acid in the P site with the amino acid in the A site. and breaking of a bond between the tRNA and the amino acid. And then the ribosome shifts. It moves 3 nucleotides or a codon at a time down the mRNA, continues those processes at the P and the A site. What's E stand for? Exit. That's where the uncharged tRNA, or the one without an amino acid, would actually leave. And this process occurs again, and again, and again, and again, over and over. About 10 every second or so. Until when? What prevents this from occurring anymore? What's that? Stop codon. Exactly. Once a stop codon is reached, a release factor binds. And this is a protein factor, binds to one of our 3 stop codons and basically takes GTP and it breaks those 2 ribosome fragments apart, releasing them from the mRNA. And that's the point at which our translation is stopped. Now, the process of translation, when we typically talk about it, we're talking about just the order of amino acids. And just to remind you, that is what we call that primary structure of a protein. It doesn't tell us about alpha helices or beta sheets. It doesn't tell us about the 3-dimensional structure of the protein either. But it does at least tell us every single amino acid that's going to be in there. Okay? What we'll be practicing then with on Friday is to go ahead, Wednesday, take DNA, convert it to RNA, and then finally finish off with what's the amino acid sequence. Okay? All right. Okay, let's finish up with 2 things, because I can just sense you guys are loving today. So, grab a blank sheet of paper. It can be a scrap paper you can share with somebody else. Rip it in half, rip it in quarters, 1/6, whatever you want. All right. And your goal is to answer this question. Take 2 minutes to answer this question. All right, take a couple of seconds to finish up. I'll collect these in a minute or two and just take a look at them. You don't need to put names on them or anything. I just want to see how we're doing with this information. One last thing that I do want to make sure to address, because I know that I addressed this in the mini lecture, but just to make a point, we will come back to this. Bacterial genomes are actually organized, and what we mean by that is genes that function in the same pathway or that function in, for example, breakdown of one particular carbon source, they're grouped together. And that allows for these genes to be turned on or turned off at all the same time. For example, where is it at? Up here in the top corner is the lac operon. The genes lac I, Z, Y, and A, they all control for metabolism of the carbon source lactose. If an organism has this operon, it's capable of fermenting lactose in most cases. Okay? So if an organism can ferment glucose, and if it has this operon, it can ferment lactose. So we'll talk about the regulation of those a little bit later. Last announcement, or one more takeaway point again today. Structural differences between bacteria and eukaryotes and how that contributes to differences in the flow of genetic information. If you're having trouble with this information, I advise you to go back to old notes, look at the book, or Julia's not going to hold a formal review session this week, so contact her if you want. And then finally, on Wednesday, there's going to be somebody else in class here as well. I don't know if anybody knows Tim Kuman, but he's coming to watch me, not you. So just so you're aware ahead of time. All right. Come bring those up to the front. I will hand back your video rubrics in the meantime.